Horseshoe crabs, or more specifically the Atlantic horseshoe crab in this case, is indispensable in both the pharmaceutical industry and for the Atlantic coastal system. This is due to their spawning habits, which occurs once a year under the full moon. They'll gather to breed on the Atlantic coast. They'll lay batches of eggs, which are eaten by migratory birds like red knots. The red knot is endangered and it travels from Chile and South America all the way to Arctic breeding grounds up in the north, only stopping once or twice. Delaware Bay being one of the most important stops and also the largest spawning area for horseshoe crabs. Other species like the vulnerable diamondback terrapin, coastal fish, and other birds also rely on horseshoe crabs for their diet. The pharmaceutical industry requires horseshoe crabs because of their unique immune system. Horseshoe crab blood is blue because they have oxygen carrier proteins, which also contain copper and turns blue when exposed to oxygen. Their immune system is made up of amoebocytes, which travel and will detect negative gram bacteria or any pathogenic bacteria. And how it functions is when amoebocytes come in contact with that pathogenic bacteria or bad stuff, it'll release clotting factor or granules of clotting factors, namely coagulagen, which then forms a gel-like, semi-solid gel-like substance onto the pathogenic bacteria, isolating it from the rest of the horseshoe crab's immune system. So pharmaceuticals have taken advantage of that by basically isolating the amoebocytes from horseshoe crab blood that is harvested, centrifuging it, and then combining it to distilled water so that the cell walls burst and the lysate or the interior of the cell comes out, and they get to purify and isolate all that good stuff into a substance known as limulus ambocyte lysate, LAL. So, ever since it was FDA approved in 1977 for endotoxin testing, the pharmaceutical industry has went from pyrogen tests, which is like mass injections of medical substances like vaccines or anything that comes in contact with human spinal fluids and blood, by injecting it into bunnies, and if they die or get diseases, then we know it was infected and we don't use it. But it's much more costly and inefficient in comparison to LAL, where you just need to combine it with whatever you're testing. And if you see clotting or color changes, then you know that endotoxins were present in the sample. So overall, there have been some alternatives, namely through genetic research, isolating the limulus factor C, clotting factor C, from horseshoe crabs, that has become much more synthetic. And overall, that's the basics of horseshoe crabs.